In early 2020, the world held its breath. A new virus, SARS-CoV-2, was spreading at an alarming rate. The traditional timeline for creating a vaccine was 10, maybe 15 years. Yet, in under a year, we had not one, but multiple effective vaccines. To many, it felt like a miracle, but it wasn't. It was the result of decades of dedicated, often overlooked scientific research coming to fruition at the exact moment humanity needed it most. Today on G Fun Facts Online, we're pulling back the curtain on this incredible scientific achievement. This isn't just the story of the COVID vaccine. It's the story of how science hacked the code of life, ushering in a golden age of vaccinology that will change medicine forever. To understand just how revolutionary this speed was, we first need to look at the old playbook. For over a century, vaccines have been a cornerstone of public health, built on ingenious but slow methods. You had live attenuated vaccines, like for measles, which use a weakened version of the live virus to provoke a strong, lifelong immune response. Then there were inactivated vaccines, like for polio or the flu, which use a killed version of the pathogen. Safe, but often requiring boosters. The development process was a slow, rigid, and sequential march. First, years of basic research to even understand the enemy. Then, preclinical testing in labs and animals. Only then could you begin the three-act drama of human clinical trials. Phase 1, with a few dozen people for safety. Phase 2, with hundreds to check the immune response. And the massive Phase 3, with tens of thousands to prove it was both safe and effective compared to a placebo. After all that, you'd submit a mountain of data to regulators like the FDA and wait. And only after getting the final green light would a company risk the massive investment to build a factory. This 10 to 15-year process was safe and reliable, but it was no match for a fast-moving 21st century pandemic. A new playbook was desperately needed. The secret to the new playbook, the key to its incredible speed, is a concept called platform technology. Think of it like building a car. Instead of designing a brand new car from scratch every single time, imagine you have a standardized, pre-approved chassis, the base frame of the vehicle. To make a new model, you just pop on a different body, a new engine, and change the interior. The core is already tested and understood. In vaccinology, a platform is that chassis. It's a core delivery system that is proven to be safe. To create a new vaccine against a new virus, scientists don't have to start from zero. They just plug in the specific genetic information from the new threat. This modular plug-and-play approach is the single biggest game changer, and the COVID-19 pandemic was its global debut. At the very heart of this revolution was a technology that turned our own bodies into on-demand vaccine factories, messenger RNA, or mRNA. The concept is beautifully simple because it hijacks a process our bodies are already doing trillions of times a second. Inside each of your cells, your DNA holds the master blueprints for everything, but it stays protected in the nucleus. To build a protein, the cell creates a temporary, disposable copy of a specific gene's instructions. That copy is messenger RNA. It travels out of the nucleus to the cell's protein-making machinery, the ribosomes, which read the instructions and build the protein. After a few hours or days, the cell breaks down and recycles the mRNA. Its job is done. mRNA vaccines harness this natural system with surgical precision. The process begins not with a virus, but with a computer file. When the genetic sequence of SARS-CoV-2 was published online in January 2020, scientists immediately identified the gene for the spike protein, the key the virus uses to unlock our cells. They didn't need the virus, they just needed its code. Using this digital code, they chemically synthesized the mRNA for just that spike protein. Now, mRNA is incredibly fragile. On its own, it would be destroyed in the body in seconds. This was a major hurdle for decades. The solution was to wrap the delicate mRNA in a microscopic bubble of fat called a lipid nanoparticle. This bubble acts as both a protective shield and a delivery vehicle. When injected, these nanoparticles fuse with our cells and release their mRNA payload. Once inside, your cell's ribosomes read the instructions and start churning out harmless copies of the spike protein. Your cell then displays these spike proteins on its surface. Your immune system spots these foreign proteins and screams, "Intruder!" It then mounts a full-scale defense, creating targeted antibodies and, crucially, a memory in the form of specialized B cells and T cells, ready to fight if the real virus ever shows up. And what happens to the mRNA instructions? They never enter the cell's nucleus, they never touch your DNA, and within a few days, the cell naturally breaks them down and discards them. The message self-destructs. This technology seemed like an overnight success, but it was built on decades of foundational research by pioneers like Katalin Karikó and Drew Weissman, who figured out how to make mRNA stable and non-inflammatory. It was a platform that was tested and refined for years against viruses like Zika and influenza, waiting for its moment. The advantages are stunning. Moderna designed its vaccine candidate in just 2 days. The first clinical batch was ready in 42 days. That's digital speed. The other star of the rapid response was viral vector technology. If mRNA vaccines are like sending an email with instructions, viral vectors are like using a Trojan horse. This approach uses a harmless, modified virus as a vector or delivery vehicle to carry genetic instructions into our cells. Scientists start with a common virus, like an adenovirus which can cause the common cold. They then genetically engineer it, stripping out its ability to replicate or cause disease. They effectively disarm it. In the space where the harmful genes were, they insert the genetic instructions for our target, in this case, the DNA blueprint for the SARS-CoV-2 spike protein. When this vaccine is injected, the harmless vector virus does what viruses do best. It infects our cells and delivers its genetic payload. The cell's machinery then reads this DNA blueprint, transcribes it into mRNA, and then translates that mRNA into the spike protein. From there, the process is the same. The immune system sees the spike protein and builds a powerful defense. Like mRNA, this platform wasn't new. It had been honed for decades and was successfully used to create an Ebola vaccine. This gave scientists huge confidence in its safety. Viral vectors have key advantages. They are known for generating very powerful and long-lasting immune responses, sometimes with just a single dose. They're also more rugged than mRNA vaccines and don't require ultra-cold storage, making them easier to distribute globally. The main challenge is that because the adenovirus vectors are common, some people might have pre-existing immunity to the delivery vehicle itself, which could potentially reduce the vaccine's effectiveness. The third major approach was a high-tech update on a classic, the subunit vaccine. This is the ultimate in precision engineering. Instead of using a whole virus, you use only a specific, purified piece of it, the subunit, that's necessary to trigger an immune response. The modern process is a marvel. Scientists use recombinant DNA technology. They take the gene for the target protein, like the spike protein, and insert it into something easy to grow in a lab, like yeast or insect cells. These cells are then turned into living factories, churning out huge quantities of the pure protein. This antigen is then harvested, purified, and formulated with an adjuvant, a substance that helps kick the immune system into high gear. An even more advanced version is the virus-like particle, or VLP. Here, scientists produce the viral structural proteins, which then self-assemble into a particle that looks identical to the real virus on the outside, but is completely empty on the inside. It has no genetic material. This imposter virus is incredibly effective at triggering a powerful immune response. The HPV vaccine is a prime example of this successful technology. The main advantage here is exceptional safety, as there is zero chance of infection. So we have these incredible platforms, But how did we know what plugs to plug in? The starting pistol for the entire vaccine race was fired on January 10, 2020, when the complete genetic sequence of the SARS-CoV-2 virus was published online. This single act, powered by the fields of genomics and bioinformatics, enabled researchers worldwide to start designing vaccines instantly, without ever needing a physical sample of the virus. This is a revolutionary concept called reverse vaccinology. Instead of the old method of growing a virus and breaking it down to find useful bits, scientists now start with the digital genome sequence and work backward. Using powerful computers, algorithms sifted through the virus' entire genetic code, predicting which proteins would be the best targets for the immune system. The spike protein was the obvious candidate. Computational tools then mapped the specific regions on that protein, called epitopes, that antibodies would most likely latch onto. AI-powered software even predicted the protein's 3D shape, which was critical for engineering a more stable version for the vaccines. This digital-first approach saved months, if not years, of exploratory lab work. But even with revolutionary designs, the 15-year timeline was still the enemy. To beat the clock, the entire development and regulatory process was reinvented. The key was shifting from a slow, linear sequence to a massively parallel effort. Instead of waiting for one clinical trial phase to end before starting the next, they overlapped. Planning for Phase 3 began while Phase 2 was still underway, but the boldest move was at-risk manufacturing. Governments and organizations poured billions of dollars into mass-producing the most promising vaccine candidates, while they were still being tested in Phase 3 trials. This was a huge financial gamble. If a vaccine failed its trial, millions of doses would have to be thrown away. But if it succeeded, those doses would be ready for immediate distribution, saving months of delay. Finally, regulatory agencies like the FDA used accelerated pathways like the Emergency Use Authorization, or EUA. It's crucial to understand, this was not a shortcut on safety. The vaccines went through the exact same rigorous Phase 1, 2, and 3 trials. The difference was that regulators reviewed the data on a rolling basis as it came in, instead of waiting for one giant submission at the end. They prioritized it above all else. This combination of parallel trials, at-risk manufacturing, and rolling reviews was the logistical masterstroke that compressed a decade of work into less than a year. This triumph over COVID-19 wasn't an endpoint. It was the start of a new chapter. The goal now is the 100 days mission, the ability to develop a vaccine against a new pandemic threat within 100 days of its discovery. But beyond pandemics, these powerful technologies are now being aimed at some of our oldest and most difficult enemies. Researchers are using mRNA to create personalized cancer vaccines. By sequencing a patient's tumor, they can create a custom vaccine that teaches that person's immune system to hunt and destroy their specific cancer cells. The ability to precisely tune the immune system opens doors to treating autoimmune disorders and even life-threatening allergies. And the innovation isn't stopping. Scientists are developing self-amplifying RNA vaccines that could require much smaller doses, and painless microneedle patches that could make immunization as easy as applying a Band-Aid. The story of rapid vaccine development is the story of modern science at its very best. It's a narrative of foresight, innovation, and collaboration. The legacy isn't just the billions of lives saved. It is the dawn of an era where vaccinology is a versatile, dynamic tool that can reshape human health, offering profound hope for the challenges of tomorrow.